look at selection in population variation data. So there's 5 different methods that we look at. I will start with the hardest one. This is the one that most people get scared of and don't understand. But you already know enough to understand this. So if you look at this graph here, this is before selection. And these are chromosomes in a population. So this is chromosome 1 of individual 1, chromosome 1, individual 2, chromosome 1, individual 3, and so on. And this is the one that gets selected. The other way of looking at it is also to see if how your variation between the sites is distributed. Do you see you have a mutation here with out of 1, 2, 3, 4, 5, 6, 7, 8, there's 5 out of 8. Okay, that's a common mutation. You also have a rare mutation, it's 1 out of 8. You also have 2 out of 8, and then you have 4 out of 8. So 5 out of 8, 2 out of 8. So one mutation has 80%, one has 50%, one has 20%. A distribution of how frequent these mutations are in the region is called a frequency spectrum. So this is a frequency spectrum. These are rare mutations, these are common mutations. There is some type of a spectrum of rare to common mutations that's common across the genome. You can expect some of them be rare, like this one, some of them be common, like this one. And with time, different mutations are at different stage of increasing or decreasing in frequency, so the spectrum should be, this is about the shape you should see. Now, the opposite is fixation. In fixation, you have everybody the same, which means if you look at the mutations, well, this is actually not a mutation site at all, because this is all fixed. Because everybody's similar, the only mutations that you can see are the ones that, which is rare, would recombine in from another chromosome, or happened since the selection occurred. Because there is some kind of mutation rate, right? And some mutations will just happen, they will occur. And these will be rare, because they can't happen across the whole population. They will all happen once, on one chromosome. So most of the mutations that you will see after selection are rare. So this is selection. Selection means no common mutations. You won't see these. You won't see variation in different sites. So that compared to this. Your selection is the background. Most of the genes will be like this. Your selected genes will be like this, and this is what you're going to see. And this is called Tejima's D. Anybody heard about Tejima's D? Everybody who heard about Tejima's D was terrified because they did not understand what it meant. All of Tejima's D means is that versus this. So here's different scenarios of Tejima's D. So here is a locus with no selection at all. Okay, no selection at all. And you can see there is some rare, there is some intermediate, and there is some common mutations. Right? Here is the spectrum. Common, rare are the reds. Rare, 1, 1, 1. Every time you look across these chromosomes, this is the only chromosome that has that mutation. This is the only chromosome that has that mutation. Now, the purple are common. You have at least 5. Okay. A selection happens on this. Okay, a selection happens on this. This selection happens somewhere here. Now, most of the mutations now are red. Most of the mutations are red. They happened after selection, and because they happened after selection, they can only happen in one chromosome. And it is not enough time for them to spread across population. So the spectrum frequency that you have is that. So do you see this versus this?
And how do you tell this situation from bottleneck? So if you have the bottleneck, you
very good question. That's an excellent question, because that's exactly what was the problem with the GMUS-D before genome studies. Because if the bottleneck happens, then you would expect every gene in your genome to act exactly the same way.
Right.
But if selection happens, you expect only certain loci to act in that way, and so you compare your gene to the rest of the genome.
Yeah.
So you can't do Tajima-D on one gene. And people have done it until now because there was no genome data. With genome data, you don't just look in one place, you look across the whole genome. And this is what you're going to see.
People are still doing it.
Oh, people still do it.
Yeah, and concerning the more complex situation when half the population had a bottleneck and say they're not trying to hybridize with somewhat more.
Half of the population had a bottleneck?
Well, not half, but some part. Yeah.
Yeah, it's a more complex. So incomplete sweeps are harder. Okay? And incomplete sweeps are harder, but still, they will hopefully be above the threshold that we'll see. But let me show you what the GMUS-D actually looks like in the real data, because we're looking at all this. This is still theoretical. But when you do this in the real data, it looks something like this. So here is this is called night sky plot or Seattle plot. And basically what they show here, if you have a homozygote that's common, it's blue. These are chromosomes, and these are loci. So here's locus 1, locus 2, locus 3, sort of like in GWatch. And there is a red here is the heterozygote, okay? And the yellow is a rare homozygote. It's a homozygote that hasn't been observed much before, but it's observed in this population. So what you will see is a population that's not selected has all kinds of variation in it. Remember frequency spectrum? There it is. So some of these loci have one heterozygote in them. Some of these loci have 5. Some of these loci even have a homozygote for the rare, which is right. That means that they're pretty variable, right? And some of them have none. So you have a full frequency spectrum. The population here that's selected has no common variation. It looks like there's some hitchhiking going on, right? Because this is rare homozygote. It's probably sitting on a chromosome that's got selected, and then it's got some, one heterozygotes. These are new mutations that start getting incorporated. So the spectrum here is only rare. Spectrum here is all kinds. So that's a signature of selection. Now, if we just looked at that and we say, well, there's a bottleneck here, right? But this signature of positive selection is a negative to GMUS-D. Negative to GMUS-D means you have no variation, right? So here's a negative to GMUS-D in this region. So each one of these loci shows negative to GMUS-D. And we can see how different that is from the background, because most of the genome on this chromosome has a positive to GMUS-D. So this is really what you're looking for. And if you look at that other population, that's the background you see. So the background you see everywhere is forest, 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 forest. Forest, 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 lion, forest, forest, forest. That's how you tell between the bottleneck and selection. Okay. But why do we see? I started with the hard one, so you catch it. Now let's go the easier ones. So what we really look for again is reduction in variation. And why make it so complicated to look at spectrums if we just can calculate average heterozygosity?
What is states for?
Huh?
Digimus, what's
Tojima is the name of the guy, of a Japanese researcher who made the equation. Is he alive? No.
I think he was one of Nave's students. Yeah. Well, I could be wrong. I just don't remember. It's been around forever.
It's been the scariest formula in population
It was in the
genetics.
70s. That he
It was in the 70s.
Yeah.
So, should have shown that before. So, heterozygosity could be used as a substitute for genetic diversity, just by saying, okay, if you have more heterozygotes, then you should have more diversity. If you have not many heterozygotes, that means everybody is homozygote. So everybody is not different. So heterozygosity is used as a proxy to explain genetic diversity. And remember, we talked about that hitchhiking and how the chromosomal part that gets selected every generation, the variations being added to it, except for the place which is selected, because you only select for one variant. So where you selected for is not variable, but everything else should be variable. So if you plot heterozygosity across the genome, it should be some value, normal value, going in every population except for the one that's selected. And so this is the pattern of heterozygosity you're looking for. Heterozygosity around selected site should be low. Heterozygosity everywhere else should be, well, whatever it is across the genome, on average. So this is this average heterozygosity, and this is low heterozygosity compared to the rest of the genome. Okay, so that is the pattern that we're looking for. Can we see these kind of patterns? I specifically did not in the paper. By that time, Steve and I have actually written a paper and looked for decreased heterozygosity in human genomes, but we didn't want to use our own examples to say, hey, we want to promote our own study. So we promoted somebody else's study. So this is not our data. Actually, none of this is our data here. So here is an example from another paper. And by the way, in these slides, you will always see, I always put the paper from where I got this from. So what these people did was to look at heterozygosity in a scan across all the chromosomes between 4 HapMap populations, 4 human populations: African, Chinese, Japanese, and European. And then they plotted an average heterozygosity across all the chromosomes, and you know, the opposite of heterozygosity is homozygosity, so you can flip it either way. So here is the highest reduction in heterozygosity or highest homozygosity across these populations. And they said, okay, we want to look under this peak. So this is critically different, and it's very different than everywhere in the genome. So we want to look under this peak and see what we can see. And what you can see here is in 3 populations out of 4, heterozygosity keeps bouncing up and down along around some kind of an average value. An average value is between 20 and 30%. That's an average value across the human genome. And do you see there's this bouncy effect. So it goes up and down. Except in one population, in this case in Europeans, this heterozygosity goes to 0 in this region, and then goes up and starts bouncing again.
What was the region?
What's the region? Right. Well, the region here, I'll just tell you, the region is SLC24A5 is the one involved in skin pigmentation. So that's one of the regions, one of the genes that makes Europeans have not so much pigment.
So it's homozygous, right?
It's almost homozygous across the whole population.
It's figure 4 in a paper I sent out on a Friday for you to read.
Yeah.
Okay, so the other thing that you might see from here is that there is also another way of looking at this. Okay, there's a decrease in heterozygosity, but there's also, you can see there's a difference in how much diversity there is here to how much diversity there is in a general population. If you put all these people together, right, this would be variable versus not variable. Do you know of a population index that describes this difference? What is it called? Not Klaus. It's called FST. FST, fixation index, right? Invented by Sewell Wright, describes exactly that.
And reinvented
And
by Jim Crow, Dick, and Eric Lander, and
yeah,
a whole bunch of
and
people.
many of those scans across populations. It's exactly that. You look at one population versus the other population, and you see if these populations, variation in these populations are different from each other. And as you remember, that the signature of selection is a reduction in variation, and one of the populations gets selected, that the difference in variation between them should be large. Okay, this is just a bunch of words. People probably just say, okay, FST, I don't want to understand this. But I can show it to you in a very simple
Why
way.
don't you define HT and HS?
I don't want to throw them off, but HT is heterozygosity in the total population. HS is heterozygosity in the subpopulation. And HI is heterozygosity among individuals. So it's you calculate it from observed in T, and I in expected is an I, and S is each subpopulation separately.
What is
And
T?
T is total.
Total. And
T total, S is subpopulation. So it's difference between heterozygosity if it was calculated as an average between subpopulations, between this one and this one, and a heterozygosity as if it was calculated if this population was 1.
But
that's mathematics.
Sorry, I don't understand why you multiply these terms 2Q1, Q1 and divide it by 2. It looks reasonable to take
Well, this
square root.
is HS. So
Yes, I
HS
understand,
means
but,
this one plus this one. You calculate them separately and then take an average of them. Because this then would be a total variance as an average of 2 subpopulations. That's what HS
Yes,
is.
but in this case it's reasonable to take square root but not divided by 2.
Maybe there are plus between the parentheses.
Or plus maybe.
I'm sorry, that is plus. Very good point. Yes, that's plus. Yes,
Oh,
that's not-
okay.
Yeah, that's a plus.
Good point.
Let's go. Let's go. Yeah, that's a plus.
So it's a sum of
It's
the
a sum
heterozygotes of the two populations divided by 2.
That's
So
what I
it's the
meant by
average
average.
between the
That's
two.
what I meant by average. But thank you for pointing it out. There's just a miss.
So Sergey would take the square root. Good idea. I like that.
Well, the geometric mean, and
So
then you take,
you'd put an exponent over that, 1/2 over there. Yeah. All right.
Alright, so,
Taras, why don't you check your equations? Okay. Go ahead.
Did you make mistake in your paper or you just in slide?
no, I didn't put that in there. I put it here for, I put it here so I could explain it. And if somebody asks a question, I can point out that I do understand.
So it's only in slide, it's good.
But again, you know, this is my problem. I'm not as good with formulas. I'm good with pictures. I'm going to show you a picture that explains the formula.
I think it's important because FST is thrown around a lot. It's a measure of differences between population, and it was originally invented by Sewell Wright, but there's variations about how it does. So Taras has just explained a little bit about one way to do it. Yeah.
And sorry about that pause. I will put it back in. So here is the two populations that have variation. Both of them have variation, but this variation in population here and variation in population here are mostly due to differences between individuals, and they're not really due to the differences between populations. They're not certain places that are constantly the same in one population versus certain places that are constantly same in the other population. It's just random. Okay? This means that FST is small. FST is a part of variation that shows a difference between two populations. So the FST is small here. That's most of the variation, that black, is a random variation between individuals, which is not what we're interested in. Okay? But in the selected locus, remember, everybody's going to be the same. So in the selected locus, then there will be consistent difference in this population from this one. So in this place, you will always have this allele, and you might or might not have it in this population. That contributes to a larger part of population differences, right, versus individual differences, which means the part of FST here is higher. In this case, it's 60%. That's all it is. Okay, so FST goes up means the populations are consistently different from each other.
So what is an empty circle? Is it heterozygous or another?
No, this is different allele.
Different allele.
Okay, and so again, if you do it in one locus, you might be misled because if you have a bottleneck, it will give you the same effect as selection. But if you do it across the genome, you can evaluate one place against the other. And here is a paper was rat tags on sickleback fish, which used to be not a model organism. Like in the last 3 years, it became a model organism. People are now publishing so much on sickleback fish, and that's because the genome is known. And they started talking all kinds of cool stuff about sickleback fish.
Well, it was a model organism for about 10 years, actually.
Well, okay, 10 years.
Because of Dolph Schluter and doing some interesting breeding experiments in British Columbia. And then, you know, that built up and spurred, actually.
Yeah, and it's been amazing.
This organism
Yeah.
to actually look for the genes that control the phenotypic differences.
And you guys know what the phenotypic difference is in sickleback fish. So it's one of those fishes that can go from freshwater to seawater and back to freshwater. And in seawater, it has more predators, so it evolves these big armor on it. So it has spikes. And if it goes to freshwater, it doesn't need that. So selection is relaxed on that.
And it's also a great example of sympatric divergence, because these fish, when they invite freshwater lakes, and this has been replicated in the laboratory as well as in the wild multiple times across British Columbia, you have what are called pelagic fish and benthic fish, and they diverge in their morphologies and their behavior, and that happens over a relatively short period of time. And we now know, because of the genome, we have the genome of these fishes, how that happens genetically. So it's a fantastic study system.
Right.